Thank you for the kind introduction and the chance to speak here while I'm in New York. So this is a picture of the Venture Institute. Thank you very much. In La Jolla, California. It's actually on the UCSD campus and is just above Scripps Aquarium and overlooks La Jolla Cove. Aside from being a beautiful building on a beautiful site, it's also the world's first carbon neutral research building. You can get carbon neutral for office buildings now relatively easy, but doing for research buildings is extremely difficult, and probably about 500 unique types of engineering went into this building to make it so it generates its own power and uses it very efficiently. So research going on at the Venture Institute has driven things in lots of directions, all around trying to answer fundamental questions about DNA and whether it contains all the necessary information for cellular life. And this applies from everything to single-cell bacteria all the way up to the human genome. And a corollary of that can we predict a phenotype, phenotype from the genotype? What's nature, what's nurture, what becomes predictable in the future? The first big leap took place when we started to what I call digitize biology, reading the genetic code, converting the ACs, Gs, and Ts into ones and zeros in the computer. And this happened for the first time with a whole species a little over 20 years ago. It was in June 20 years ago this summer, where we sequenced the genome of Haemophilus influenzae that causes meningitis and causes ear infections in kids. And it was a combination of new mathematical algorithms and strategies that put this together to become the first genome in history, even though there had been several people working with government funding for close to a decade on other genomes. 5 years later, this led to the formation of a company, Celera Genomics, where we sequenced the first draft of the human genome, substantially larger than the first bacteria. Took 9 months, 350 machines, and about $100 million. It wasn't a highly replicable event because of the cost and scale. The only thing it seemed like a bargain was compared to a $3 billion federal program that it was competing with. So we've been accumulating lots of biology in the computers as ones and zeros, and one of our challenges was to go the other way physically. So the challenges are to read those ones and zeros and understand biology, and the other was to see if we could recapitulate biology. So we developed tools to actually rebuild the genetic code from 4 bottles of chemicals, starting with the information in the computer. Now this was done initially as a test with the genome of Phi-X 174. It's about 5,000 base pairs. We chemically made that genome, injected it into E. coli. E. coli read this synthetic piece of DNA, started making proteins. The proteins self-assembled. They formed the Phi-X 174 virus that kills E. coli. And you can see the clear spots on this lawn of E. coli where the virus has killed it, then burst out of the cells, infecting more cells. So we called this a situation where the software was building its own hardware. We just put in a piece of chemically made software into these cells and got these infectious viral particles produced as a result. We really wanted to scale up substantially, and there was a good 5 or 6 years between the last slide and this one, where we've developed tools to synthesize and assemble a 1.1 million base pair genome. The DNA synthesis and assembly tools were very primitive when we started. The machines that write the genetic code are error-prone, so we developed ways to correct the errors. But we started by making this, designing first and then making 1,000 base pair pieces. We could take 10 of those, assemble them to make 10,000 base pair pieces. Then we could take 10 of those to make the 100,000 base pair pieces. And by then, you're getting to pretty large pieces of DNA that get hard to handle and clone in the different cloning vectors. What we found is we could just put these into Saccharomyces cerevisiae, and because of its homologous recombination, would simply stitch these pieces together for us, and then we could isolate the bacterial genome that we synthesized from yeast and transplant it. Transplantation was harder to work out. We had to develop ways to actually move these whole chromosomes around. DNA is very brittle, and if you try and pipette it, it breaks, so we have to move things around in gel blocks, but we found a way to insert the genome into a recipient cell. And so now we have an unusual situation, but not totally unique in biology, where we have a single species by its phenotype and two sets of genetic instructions. Well, we designed the synthetic cell, the synthetic chromosome, so when its genome started to be read, it would produce a number of proteins early on. These are the restriction enzymes that recognize the other chromosome in the cell as foreign DNA and chewed it up. So now we have a very unusual situation of the phenotype species of one organism and the genetic instructions of another. And what happened over a relatively short period of time is the new genetic instructions took over the cell, replaced every molecule in it, and became what was coded for. And that's what we reported in 2010. These are the first synthetic cells with the definition of being everything in them derived from that synthetic chromosome. This is key because some people like to talk about epigenetics. These cells have tons of epigenetic phenomenon, but they all derive directly from that synthetic piece of DNA that was put in. So help prove the linear coding codes for all these parameters. And I think it helped prove that life is a DNA software system, and also just by changing species, by changing the DNA, it became an important principle that's guiding this field. Now, we're doing this and forcing this to happen on sequencing large numbers of genomes in the ocean. We found multiple species that had a range of chromosomes that were very different from each other. And so they clearly acquired them either from the environment or from fusing with cells. And as long as the restriction enzymes were compatible, they didn't get chewed up and destroyed. So in fact, we've had giant leaps in evolution from species taking on a whole new set of chromosome material, adding literally thousands of traits in a single event versus this notion that we're changing things one point mutation at a time. So progress over the last 5 years has been slow and difficult because we've been basically trying to design a species in the computer for the first time and testing the limits of our knowledge. So we started with synthetic 1.0, which was largely based on plasma mycoides. And we wanted to design a minimal genome. This has been the goal since 1995 when we did the first 2 genomes, just trying to ask what the basic components of life were. We did this through basic design, designing what we thought should be in a cell. We made those components and we couldn't get life. So basically we had to systematically add back pieces to find out what was wrong with the design. And basically the design was good except for about 10% of the genome, which are unknown genes, or genes of unknown function, that if they're not there, you can't get life. So it's very difficult to design something de novo when you don't know what 10% of the pieces do. My late uncle was lead designer for the Boeing 767, and I was on a book tour that he was on in Seattle, and I said, imagine if my uncle designing the 767 didn't know what 10% of the parts did. And he shouted back from the back of the room, what makes you think we knew? And in fact, the process of aviation has been one of trial and failure. Plane falls out of the sky, it's intensely examined to see why, and that part corrected next time. So that's more or less the process we've gone to for the last 5 years, and we have a paper pending right now describing the smallest genome of a self-replicating organism, and it was completely designed in the computer with the addition of random experiments to add back the missing pieces. We're also working on defragging the genome. If we're going to do things by design, it'd be nice to have things much better organized by function. And we've been able to do this, but just to show you how complex it is, evolution really scrambles things with all the genetic changes. So this is just taking a 1/8 of the genome, and you can see all the rearranging we have to do to organize genes by function. The good news is this actually works, and so you can have a highly rational design and have a highly ordered genome, and it works just as well. And we ended up with these scrambled genomes with all the trillions of changes over billions of years. So this is progressing nicely, and we're going to be at the point very soon with this very first designed synthetic cell. The other approach we've been taking is from the other end of the scale of starting with humans, and so trying to understand the human genome. But I think working on a minimal cell helps put that in context, because if 10% of the genes in the smallest self-replicating cell are unknown to science, when we look at the 20,000 something odd genes in the human genome, it's much more humbling. In fact, we only know a tiny fraction right now of the human genome. And there's a lot of different approaches to this, but it was exciting when we sequenced the first human genome and at least narrowed down how many genes there were and a few other discoveries. But it's been a paucity of new breakthroughs in the last 15 years, except on the technology side. So a number of things have come together. We're sort of waiting for this threshold. Obviously, the DNA sequencing has changed so that now for under $2,000, we can rapidly sequence a human genome. So going from 100 million to $1,500 or so is a huge change. The computational power has changed equally. We built a $50 million computer in 1999 to assemble the first genome. It was 1.5 teraflops. For about $100, you can buy a teraflop card for your PC now. And with cloud computing, we can use literally thousands and thousands of nodes in a variable fashion. So change in sequencing technologies, major changes in computational power, and then we're combining that with machine learning approaches, which are changing how we put things together in the computer. So we started human longevity just about 2 years ago. I hired Franz Ock out of Google. If you've ever used Google Translate, it's because he, with a small team using machine learning, developed ways to translate over 100 languages into each other. So I convinced him we had the ultimate translation problem. So we built the largest human genome sequencing facility, and just during this setup phase, we've already sequenced 20,000 complete human genomes, where we have phenotype and or clinical data on all of those individuals, because sequence alone is not useful without all that other information. These are the machines. So each one of these $1 million machines is 1,350 times all of Celera 15 years ago. So it's a massive change in technology. But we're collecting all this different information along with the human sequence, clinical records. We're doing extensive phenotyping, including imaging that I'll show you. We are working with Metabolon to measure over 2,400 different chemicals. We sequence the microbiome and combine that with the germline. So it's a combination of creating these complex databases that can put data together in the cloud that has not been linked together before, and using machine learning to try and sort out different pieces of it. I'll give you some examples and show you what we're doing, but looks like many people in this audience fall somewhere in this range. If not, you will soon. So if you're between 50 and 75 years of age, if you're a male, you have about a 30% chance of dying during that time period of your life. About a third of that is from cancer and a third from heart disease. You can see if odds are better. If you're a woman, you only have about a 20% chance of dying. Increased percentage of cancer, slightly less cardiovascular disease, but roughly 2/3 of your risk of dying is either heart disease or cancer. Then you can see there's a wide range of other diseases. Our view of human longevity, if we can impact any of these areas with early detection, prediction of risk, elimination of risk, or early detection and treatment, it will definitely affect your longevity. So we're combining all these different types of data into subject databases, and then we're combining those subject databases into large ones. Here's just some of the phenotype data that we're collecting at our health nucleus, and I'll go through some of these so you don't have to read them all. So the microbiome, there's several different versions of this, but we have more microbial cells than we have human cells. We primarily do from the human gut and sequence that, but we can do from skin, from the vagina, from the oral cavity, and really get a comprehensive characterization. There's limited number of things right now that can truly be diagnosed with it. We're more trying to correlate it across the board with the information we get on metabolome and the genome. And so with the metabolome, we can measure all these repertoire of roughly 2,400 human chemicals. But after a meal, we find around 500 chemicals in somebody's typical bloodstream. 60% of those are from our own metabolism, what we generate. 30% are from the different species you consume in your diet. But about 10%, or about 50 chemicals circulating through your brain right now, are from bacterial metabolites of your dietary chemicals and human chemicals. So because when we sequence the microbiome, we sequence the complete set of genomes of all the microbes, not just 16 sRNA like most, we can now create metabolic pathway associates where we can find the pathway in the microbe, the pathway in the human, and correlating those with the metabolites we find in the blood. Some of the associations are getting extremely interesting looking at these in common. We're using extensive imaging for basically the first time measuring all these different things in theory on healthy people. This is our new 3T MRI that has protocols that only we in GE Research have. Right now it takes about 2 hours to go through it, but we measure a lot of things. But those of you who don't know anything about MRI, there's been a big change just in the last year or two with diffusion-weighted imaging. And I'll show you what that means, but just simply by measuring the water differences in different parts of your body, we can get totally unique imaging and detecting cancer from normal tissue, for example. We do quantitative imaging of the brain and using a new set of algorithms called NeuroQuant. You can get precise volumes of 20 different brain regions. HLI is an unusual organization in many ways, but it's probably the only organization where people try to play one-upmanship by the size of their hippocampus, because we can give you precisely your hippocampal volume Right now, Franz Ock, the head of machine learning, has the largest hippocampus, which maybe isn't surprising. So, but with this imaging, we can tell differences in as little as 3 months and predict between imaging and the genome 20 years before you would experience symptoms with dementia. This is the kind of imaging we can get of your carotid arteries without any contrast media whatsoever. So there's no intervention here. This is just due to the water molecule movement difference in the bloodstream versus the surrounding tissues. And if there are bends or occlusions or problems, they would show up immediately. We can actually get your entire brain vasculature. And again, somebody that either had a blockage or an aneurysm, it would show up extremely clearly. So all of this totally non-invasively, so it's getting quite exciting. And because of the water differences now, tumors generally show up different than benign tissues. So this is a prostate cancer tumor that showed up that would not have been seen with earlier versions of MRI. And David Caro, one of our collaborators and colleagues, and his team published a data showing what they got from this aqueous difference imaging and how it correlates with the pathology. So we're able to get so much more information now from non-invasive imaging. The old concern of if you test, quote, healthy people, you will find just lots of incidental omas that end up not being something significant, I think is a thing of the past. And we're making very substantial discoveries in this space. Cancer is one of the areas that's easiest to address right now because we can make immediate links between driver mutations that can be used as diagnosis for that specific drug. One of the best examples is with the ALK gene. Pfizer did a large clinical trial with a new lung cancer treatment that failed. But a retrospective analysis of that clinical trial with measuring whether patients had the ALK gene translocation or not showed up about 5% of the patients with lung cancer had the ALK translocation. It's about 1 in 400,000 in the general population. So that was very telling on its own. And when you look at responses versus not responses, that small group of 5%, over 60% of them responded to the drug treatment. That got the drug approved now, and it's also the diagnostic test that you're given before you will be given the drug. Because insurance companies know, even though it costs over $100,000 a year for that treatment, that instead of giving it to 95% of patients that it won't help and might harm, they're only paying for it for the 5% that have a reasonable chance of increased or improved outcomes. In cancer, we're measuring a lot of different things. We have programs where we measure the genome of the patient. We measure the genome of the tumor at a 90x coverage. We sequence RNA in the tumor. We sequence the immune system. This is a middle-aged man that had head and neck cancer caused by HPV-16. This is sort of a new epidemic that's occurring from HPV-16 inserting in the genome and causing cancer. From this individual's genome, he had 3 major oncogenes that were mutated, 5 genes associated with his immune system that would indicate he probably had a suppressed immune system, which he did. And we found HPV-16 present in his microbiome. Just knowing those things alone, if we found that on any one of you, we would say that your chances of getting cancer are extremely high. It's more of a question of when, not if. And one of the questions is the HPV-16 vaccine that's used for young girls and boys now to vaccinate them against getting cancer from HPV, would that work as a preventative measure in individuals that already have HPV16 present. We found 25,000 mutations in this guy's tumor, 315 of those in protein coding regions. And we can take all this data into pathway data and not be limited like companies like Foundation are to just looking at a small set of mutated genes. We can find any druggable target in those pathways and come up with whole new cocktails. The other approach we can do from these 315 mutated genes, if you understand that, DNA mutations changes the protein structures. If you have an intact immune system, your immune system probably will be clearing these cancers all the time. Now, it's not a problem here in New York, at least from the vitamin D deficiency data. In La Jolla, if you go out in the sun for an hour, something people actually do, you can get thousands of mutations in your skin cells in an hour. We replace our skin every 2 weeks, so it's not a significant problem. But in all tissues, we start to accumulate DNA mutations that, if they cross a certain threshold, turn into cancer. With seeing these mutations, your immune system recognizes them and constantly clears the tumors. If your immune system can't do that, they develop into cancers. New signs of therapy are trying to find various ways to enhance people's immune system to do this. We're adding to it by making RNA vaccines designed against these altered proteins to help the immune system work against these. And we'll be trying some trials very soon. The goal is to go from within 3 weeks of getting the tumor sample at HLI to having an individualized vaccine ready to give to the patient that can be done in conjunction with other immune therapy. With our whole body imaging, the level of resolution with these new protocols is absolutely stunning. We can get exact quantitation of muscle mass and other things, and sorting through, if you have a tumor larger than 2 millimeters, we can find it. But we can get even more interesting metabolic information, working with a group in Sweden that can transfer these images so we can get quantitative amounts of visceral versus peripheral fat. So you can see in the green there on the left, that's visceral adipose tissue, which is the dangerous stuff that is very hard to measure. We can go over the liver and get an exact amount of quantitation of how much fat you have on your liver. I was going on a 30-mile bike ride with one of the world's experts on this fat, and halfway through the bike ride, he goes, you know, exercise won't get rid of that. And I said, you couldn't have told me that 15 miles ago. I wouldn't have gone on the ride, maybe. The only way to do it is actually calorie restriction reverses that dangerous visceral fat. So, but we get precise bone volumes, bone density, muscle volumes, muscle density, peripheral, visceral fat, and this is something that you can all change dynamically as you can your hippocampal volume with different approaches. This is, we do CT scanning. This is actually a CT scan of my heart. The resolution on these things is really stunning. We get quantitative information on your coronary vessels. I was pleased to see that my heart wasn't super covered with fat, but we also measure the heart with a new 4D echo system. And this is stunning. In just 20 minutes, we can get these multicolored videos. You can see all 4 chambers of the heart. We can measure all the valves, wall thickness, and then we can zoom in. You can see that's just looking right down the aortic valve. You can see the valve opening and closing. You can see if there's defects in it. And then we use Doppler shifts, and you can see whether all the blood's going out or some is coming back in if you have a leaky valve. Just early on, 2 physicians going through our program, we discovered that they, just with the echo, that they had a distended aorta. One was a heavy weightlifter with hypertension. So his first symptom would have been sudden death just from blowing out his aorta, and now he's getting that surgically repaired, and we found another one. So it's each of these tests keeps coming up with unique measurements. We can measure gait. We have a special device that records precise things about your gait, your balance, and can detect minor changes associated with different diseases and parts of your neural system. And we capture head-to-toe 3D imaging to create avatars that will help walk you through your data when we integrate all this information. We're also adding in remote sensing and testing to supplement what we get from all these components in-house. And then we take all this into machine learning of trying to make new associations and make predictions. And eventually, perhaps with stem cells and CRISPRs, changing and editing the code. One of the studies that we're doing as a test is we're trying to see if we can predict your age from the genetic code and trying to predict your exact photograph. And we've made tremendous progress from this, creating large data sets and converting that into knowledge with machine learning. For the face study, oh no, this is the aging study. You may not know this, but as you age, women lose their X chromosomes and men lose their Y chromosomes. And this is sort of just data on a number of people showing that loss. So that's one of the measurements we can do to estimate your age or biological age, is how many of your chromosomes you've actually lost. Telomeres do change, and we can quantitatively measure telomere differences in people, and with our sequencing it's more accurate than any other ones, and we can relate these to metabolic data. Been putting this together, we can measure changes in mitochondria. There's a number of other changes. So if it asked me 10 years ago how many times you need to get your genome sequenced, I would have said once, unless you develop cancer, then with your tumor. And I'm quite frankly surprised how many genetic changes there are that we can relatively precisely estimate at least your biological age, and usually that associates in most cases with age from sequencing your genome. So it does change over time. With our face study, we take complex 3D photographs of it was over 1,000 people's faces, and we generate about 30,000 measurements on the face. And then we developed a training set that took that into machine learning. We also do this with voice. We just do voice from people reading a set phrase and then free speech. And I've been quite frankly really surprised with the voice data. We're confident we'll be able to predict your voice just from your genetic code, but just from your voice patterns right now, we can accurately tell your age, your sex, and your height. This is one of the things with machine learning that brings in parameters out of correlation that seem surprising, but all our voices are proportionate roughly to our body size and volume, and height becomes a good predictor. Eye color now we can predict extremely accurately. In fact, we're better at describing your eye color from your genetic code than you can self-describe. People are inherently bad about knowing what their eye color is or other people's. So it's getting increasingly accurate at that. Skin color, we're extremely good at that as well. And so we've taken this further now to try and be able to predict faces. And ethnicity is a key part of any sort of genetic prediction, because a lot of things go in from the genome into these ethnicity predictions. But I'll just show you one example. So the top are the actual pictures of this woman, and and the bottom are the straight computer predictions from her genome. And we're having lots of these come up in correlations now, and they will improve as the data sets improve. One of the biggest surprises, we're trying to know what age does your genome code for that you would look like? As an infant, as a 90-year-old? Well, it actually seems to code for what you look like post-puberty, which I guess makes sense, sort of the adult mature form. But now we can take software to de-age or age photographs to get them to look more like the present age. But it's getting exciting as we go forward because the forensic applications of this of just from a DNA sample being able to accurately predict somebody's face, their height, BMI, and other characteristics. And here's some of the voice data of predicting sex and age, just to show you the hints of some of the things to come. So we have data from a very large number of sources, from clinical trials we're doing with the pharmaceutical industry, with trials in cancer, with just academic groups that have well characterized a unique phenotype in a medical population. We're sequencing those genomes and adding the phenotype data. So our goal is to get on the order of 1 million records by 2020. But this is probably going to happen much faster now. We just announced a month or so ago a deal with insurance company Discovery Health in South Africa and England. They have 4.4 million clients they're offering this to, and that starts January 1, and we're talking to multiple others. So there's at least a remote chance this could be in the tens of millions. And the discoveries that are starting to come out of this, I think as they roll out, I think will be exciting. But the face is important because as you get a report about you, we want you to know, in fact, that it is you. We can tell at the sequence level identical twins apart, just from the somatic variants. So each of us is truly, totally unique. And as this data comes out and the predictions about the health and outcome data, if you believe this is a description of you, that's very different than other members of your family. Family history is important, but we all get thousands of spontaneous mutations. So as much as we look like our parents, each of us have unique elements, even separate from them. So that's a quick overview of where things are going. I'm happy to take questions if there's time. Thank you.
Good. Okay. Go ahead.
Where was the first? Yeah. I think the question is, what's for lunch?
So you showed this modular DNA where you group by function, and you said it was equivalent to the case which was typically present. I'm curious if there's any gains. Is there any added efficiencies or improvements that come from that modularity?
In the microbe that we've been doing this with, nothing that's obvious yet. It's hard to measure complex phenotypes in that setting. We're much more concerned with the loss, for example, of cell division rates or something that it would be somehow deleterious by not moving all the regulatory elements with the gene. So we're trying to capture those, but a lot of those are unknown. So either we're capturing them or So there's no loss of function, which is important. Nothing's obvious yet in terms of more efficiency from things being grouped together. I think that was one of the biggest surprises. Everybody kind of thought biology was going to be highly ordered. And then I think early on, the nicotinic acetylcholine receptor that controls our muscle contraction has 5 subunits. And when they were mapped, they were all on different chromosomes. And so the notion of logic and organization went out. And the government and NIH would like to have chromosomes that just code for the heart so they can fund the Heart Institute and code for the brain. But biology doesn't work that way. But it'd be interesting as we get more sophisticated systems to try and see.
You try to understand everything which comes out of the gene in respect of phenotype. So my question is, what was the biggest surprise in respect what you cannot predict?
I think it's too early to answer that question.
Okay.
I mean, it really is. I mean, the goal is sometime in the next decade, I think we'll have enough data to try and answer the question of what's nature. And by difference, everything else will be nurture. But I think we have to finish gathering these populations. I'm more surprised by what we're finding than what we're not finding yet.
I have a...
Yeah.
Oh, go ahead.
I have a personal question. I read your bio. When you were in 7th and 8th grade, you were getting C's and D's. And now you're one of the most celebrated scientists, researchers. Your discoveries are going to change the world. So what are we doing wrong in our education system, and what should we be focused on for our elementary schools and high schools to become like you?
Yeah. So I think one of the things that will come out of this data is how different and unique each of us are. And our education system is geared for a certain subset of the population, more than not, that do extremely well with regurgitation, memory kind of learning versus understanding. And we think some of these things will correlate with physical correlations. I usually take polls of student audiences and stuff, but how many of you here have what you describe as a photographic memory? So this must be a highly intelligent group, because that's higher than even most averages. And how many of you have no visual memory whatsoever? You can't see any pictures. Okay, there's one. So that makes 2 of us. And there was a study done in the late 1800s on you have to rely on self-reporting, right? There's nobody else no other way to measure what kind people really have. The report in the 1890s of these surveys was that it was primarily scientists that had no visual memory, and they were more conceptual thinkers. So maybe if we understand people's brain types, I hated rote memorization, so I thought it was the stupidest thing ever to memorize words and then regurgitate them on a spelling test, and I refused to take the test. I'd be in deep trouble if it wasn't for spell check today, because there was a reason for doing those tests. I just didn't really like it at the time. So I think it's understanding the differences in people and making sure maybe they're slotted into the right kind of education system. People like me are sort of shunted to the side. And maybe that's good as well. I didn't have creativity driven out of me by the education system, because I was off doing creative things, building boats and having fun. So, and when I was ready for the education system, I could survive it. So, yeah.
Final question before we go to lunch. Yeah. I find it interesting that information technology made it possible for you to crack the human genome. And then you said in one of your slides that the implications of a lot of your genetic research is that humans are basically a lot of code, a lot of software. if we are through the kind of work that you're doing, and we heard from Jennifer Doudna and Emmanuel Charpentier earlier today with CRISPR, and we've heard a lot about a lot of things being code these days. It's one thing for medicine to be trying to fix things or prevent things in the existing human structure. But the implications of code are that once you understand the code, not only can you correct code, but you can write new code.
Yep.
And you're doing that with synthetic biology. What do you think the implications of the concept of us as code will lead us?
So, and I've written about this, I think it will be inevitable that we will try and change our code. But it's way too early to do it. Sort of, the question, what are we learning, what are we not learning? It's such an early point in understanding our code. And when we look at other species in biology, there's some clear warning lessons, because biologists focus in and they think they understand the function of a gene. There's no such thing as the function of a gene. We work in these vast networks. One of the nice things about machine learning, it's the opposite of the field of genetics right now. It doesn't think of a single point mutation or a single gene as causing things, and it looks across the genome, and we measure thousands of things that contribute to a trait. There's, for example, a developmental gene in fruit flies that if that gene is mutated, you end up with odd numbers of legs or wings. And so you could say that's actually just key for development, which it is. But in the late stage of development, that gene codes for a protein that becomes a key structural protein in the wings. And so until we understand the network of functions of genes, what does it really do in the human body, I think it's going to be dangerous to try and alter them. Now, there are some that are relatively obvious that we think we could make changes in that could alleviate a fair amount of suffering. Sickle cell trait, sickle cell disease, just moving those back to normal, in theory, would be a benefit. But things derive from different populations. When we look in Saudi Arabia and the Mideast, it's the highest incidence of type 2 diabetes. It's from all the inbreeding in those groups, but type 2 diabetes was a selective advantage in a Bedouin existence, and that's how it got selected there. Sickle cell trait actually protected against malaria. But when you're now not a Bedouin existence, you're on a Western diet, that same trait becomes a negative selection. And so we just need to be smart about it. We can do more, we can change more, than doesn't mean we should do it. In fact, my view is we shouldn't do it until we have sufficient knowledge. Once we have the knowledge, I think we should go at it full force.
On that note, on behalf of all of us, thank you.
Thank you.